All right, we need to get some notes on enzymes. And we're still kind of talking about chemical reactions because enzymes speed up chemical reactions. Enzymes are a globular protein, meaning they look kind of like a blob. They are the most advanced or folded type of protein. And if they're a protein, what is their monomer? If you said amino acid, you would be correct. Okay, so there are some things that are going to affect any chemical reaction, whether it has an enzyme or not. And when I say the rate of reaction, it just means how fast or slow it's going. So temperature, we saw that with the film canisters. As the temperature increases, the rate of reaction will increase, meaning that the top of the film canister came off quicker. As the temperature decreases, the rate of the reaction decreases. So the one with cold water had the lowest or the slowest time to pop the top off. So temperature will speed up a reaction. That's the whole point of cooking. When you increase the temperature, the reaction is going to happen faster. Also pH. Do you remember talking about pH with water? Well, if a reaction is in the right pH, it will go at the correct speed. If the pH is disrupted, then it can go slower or faster or not at all. And this is sometimes like if your tummy hurts, the pH of your stomach has gotten off and it's not able to break down the food the way that it wants to or at the way that it's ideal for it. And so you take Tums and that kind of helps rebalance the pH, or at least that's what I do. Other things that affect the rate of a chemical reaction. The concentration of the reactants. Another name for reactants is substrate. We're going to start talking about substrates a lot, so just know that they're synonyms. If you have more stuff, the reaction is going to happen faster. We saw that yesterday when we put 2 tablets into the canister. It happened faster than with 1 tablet. And then lastly, adding a catalyst. And a catalyst is just a special substance that is going to lower the activation energy of a reaction. And if you remember from yesterday, activation energy is that energy required to get the reaction to start. If you are a driver and you've started a car, you know you kind of flip the key a little bit farther and it has a higher energy level. And then once it catches, you let go and it kind of flips back a little bit. Exactly, you need to get to that activation energy and then you can go back. Once the reaction gets started, you don't have to have as much energy. So a catalyst is something that makes that activation energy be not quite as high. Kind of like if you were able to change a mountain into a hill, and then you were able to get to the other side faster if it's a hill rather than a mountain. You can see that I came up with that analogy because of that picture down at the bottom. Take a second and see if you can make sense out of that picture. Now, we've been talking about enzymes, and this is probably so important, so that's why I have it in bold. An enzyme is just a catalyst in living things. So an enzyme is going to speed up chemical reactions inside any type of living thing. You have thousands upon thousands of enzymes in your body. Even single-celled organisms have several enzymes in their bodies or in their cells. If you increase the amount of catalyst, or if you increase the amount of enzyme, the concentration of it, then more reactions can happen simultaneously, meaning at the same time. To a certain point, this graph isn't completely accurate. It should kind of level off at a certain point because you would also need to increase the amount of stuff or reactants or substrates in there to really get it going fast. Okay, so look at these pictures and see if you can make sense out of these pictures. I like the picture on the right the best. So look what's happening. We have the yellow thing, that's our enzyme. Our red and green are our reactants or our substrate. They go to that enzyme, and the enzyme smushes them together and makes a new substance, which is that purple goggly thing right there. So I've said this before, but I can't emphasize this enough. This is something that's really important for you to like kind of stick in your brain. An enzyme is a protein that its job is going to speed up a chemical reaction by lowering the activation energy. So it decreases the amount of energy you need, and that's going to make the reaction happen faster. All right, I don't love the way that your notebook is laid out here. So you're going to have to write the next couple of slides at the bottom of page 25. They're not really in your notes. So take your time and make sure you get everything written out clearly. There are going to be some things that I can tell you that you can skip. So enzymes can bring things together, just like we saw in that last picture. So here, this picture is labeled really well. The purple is the enzyme, and the yellow and blue are the reactants or substrate. And they are going to come together on the enzyme. And this point right here on the enzyme is called the active site. And then the active site is going to, when they join there, they're going to get smushed together and form the product. And then the product's going to leave. And look, that purple enzyme at the end after the product is left is exactly the same as it has been at the start. So it can be used over and over again. Enzymes very rarely are used up. They can also break things apart. So the bottom picture is kind of a reverse. You've got the enzyme and you've got the purple, which is the substrate. This time there's only one substrate. And then the enzyme breaks it apart into two products. Okay, so one thing that I think I would like for you to do when you finish this slide is to draw this picture down here at the bottom. The other thing that I would like for you to do that I realized just now that I don't have in here is when you draw this picture, make sure that you label that this is the active site. I'm going to try to draw it on here. I'm not going to lie, that might have been my best writing on a slide ever. So up at the top, it says enzymes are very specific. They have an active site, which I just pointed out right there. Maybe underline active site because that's an important word too, that only fits one substrate. So you have to have an enzyme for every chemical reaction in your body. Sometimes you have enzymes and their only job is to make more enzymes. Because they're very specific. They only work with one type of chemical reaction. This model down here is called the lock and key model. So looking at this picture, which do you think is the lock and which do you think is the key? If you said the enzyme is the lock and the substrate is the key, you are correct. And that key is specific to the lock. So, and we saw this on the last slide, the enzyme binds to the reaction. What really happens is it changes shape slightly, and that either breaks bonds or makes bonds form, forces them together. And then they release the product, and the enzyme goes back to its regular shape. It's kind of bendable. I think of it like Play-Doh. But so they go back to their original shape, and so they can be used over and over and over again, unless something bad happens. But I'm getting ahead of myself. This is the last thing that you're going to have to write at the bottom of page 25. And then there's only one more slide. Now you notice this in our lab, our simulation activity. There are things called inhibitors. and they are going to block the active site of an enzyme and slow the reaction down. One inhibitor that you probably know about is blood pressure medication. So you take that blood pressure medication, your body breaks it down, and these are inhibitors that block the enzymes that raise your blood pressure, so it keeps your blood pressure lower. Then there are coenzymes, and these are helper enzymes that are going to bind to the active site, and so the substrate can be broken down faster or joined up faster. Our body manufactures coenzymes. You can also take them, they can be called cofactors if they're like a metal, like iron or something like that. All right, so up at the top left of page 25, there is a box about denaturization or denaturation. This is the last thing you need to write down. There's going to be two things on this slide, but you only have to write down one of them. So the first thing, this is already on your paper. There are things that can totally warp or misshape or ruin an enzyme. And the two major ones are if it's not in the right temperature or if it's not in the right pH. The enzyme will become denatured, it'll lose its shape, and it won't work anymore. So during denaturation, the enzyme changes shape, and then the active site can't work on that substrate anymore, and the reaction doesn't work. So I don't know if you've ever been like out in a creek or something that's gotten really low. This is an example that happened to me in my childhood. And when the creek gets low, the water gets really hot and the fish start dying. And that's because the water temperature is changing the temperature of the enzymes of the fish because they're cold-blooded. They take the temperature of what's around them, and their enzymes get warped, and then they can't do their chemical reactions and they go belly up. Another one would be like with your tummy, if it gets too acidic, the enzymes aren't going to work anymore. Sometimes denaturation can be fixed, like the enzymes in your tummy if you eat or if you take some medicine, but sometimes they are warped beyond recognition, and that can cause a lot of health problems.